This is Dr. Muzaffar Ahmed Bhatt from Department of Sericulture, Poonch Campus, University of Jammu. Today we will discuss about silkworm as a laboratory tool. Definition of laboratory tool. It is used for a particular species or organism. It is extensively used in research laboratories. It has the advancement of understanding of cellular function, development and disease, and it has the ability to apply new knowledge to other organisms. The current models which are used for laboratory research are Drosophila, silkworm, xenofish, zebrafish, mouse, sea lichens, yeast, E. coli, and Arabidopsis. The characteristics which make silkworm as a good laboratory tool are: first one, it is a small insect, easy and cheap to maintain and manipulate. It has short lifespan. It produces large number of offsprings. Its development is external. It has the availability of mutants. It has lots of history, previous experiments and discoveries. Its genome is sequenced. It has homologs for at least 75% of human disease genes. It exhibits a complex behaviors. It has fewer ethical concerns. Silkworm in research. It has the early research aided in the understanding of development. It has made first link between chromosome and phenotype. It is identified for various genes and mechanisms of development. Now, recent applications of silkworms as an experimental animal. Application as an animal for primary bioassays uses several times in development. First one, to develop new drugs. It is common to use mice and other mammals to measure the biological effectiveness of chemical compounds. The associated expense, however, is large, and attempts to reduce such expenses or switch to use of non-mammalian animals are underway. When screening many chemical compounds, the use of silkworms in the initial step of experiments to measure biological effectiveness is convenient. Although examination to verify whether the efficacy of drugs in silkworms is the same as that in humans is vital. Silkworms can be used as a convenient model to narrow down the number of candidates for many chemical compounds. Utilization as a protein production system. Silkworms are utilized to obtain targeted proteins by expressing cloned genes. One utilization method obtains targeted proteins by integrating cloned DNA into genome of viruses living within the host silkworms. The transgenic virus proliferates in the silkworm body, and silkworms usually die in a few days because of the proliferation of the virus. Proteins derived from exogenous genes depend significantly on the host silkworm strain. This production system of targeted proteins has already been commercialized by some business corporations. Now, development of custom designed strains responding to requests from researchers. Many researchers apparently think that mulberry leaves are essential for rearing silkworms, and also that it is not possible to rear silkworms in winter season due to the lack of leaves. To solve this problem, an artificial diet, soybean protein as the main component, has successfully been developed. Rearing various mutant variant strains on an artificial diet, however, has proved difficult. To solve this problem, NBRP has bred a group of silkworm strains that will feed on artificial diet without problems, and has created many silkworm strains that are available all around the year. Currently, researchers who are unable to obtain mulberries can utilize silkworms for research work anytime with a commercial artificial diet. Now, summary. First one, benefits of silkworm include broad spectrum of genes related to human disease already discovered, Many successful techniques already developed, already a powerful tool in study of developmental and neurological disorders and cancer. Second one, future perspectives. Identification of novel genes functioning in disease processes, determination of genes contributing to complex disorders, exploit the flight to answer already existing questions and formulate new hypotheses. Silkworm is a most effective model because the larva of Bombyx mori has a moderate body size. This is very convenient for performing surgical operations such as removal or implantation of a specific organ. Such implantation experiments have been carried out by many investigators for understanding the function of sex hormone, molting hormone, and other internal secretions. More simplicity than vertebrate models, great complexity than yeast or bacteria models. Thanks to all.